Our next speaker is Ben Streatch. He's a master's student at Queen's University, and his research focuses on how lab conditions in animal colonies can affect indices of anxiety. And Ben, I'm looking forward to your talk.
Okay, can everyone hear me? Yeah, perfect. Okay, so hey everyone, my name is Ben Streatch, and today I'm going to be talking to you about my recent work, evidence of social buffering in rats housed in individually ventilated cages, also known as IVCs. So a little bit of background before we really get going. So historically, rodents have been kept in these open-top cages, or OTCs, and those use one ventilation system for a group of cages. So you can see an IVC in kind of the top right-hand image there. So it's really just a plastic tub with a wire lid. Recently, however, there's been a big push to switch over to these individually ventilated cages, or IVCs. And you can see one of those in the image at the bottom. And so that's actually a hermetically sealed plexiglass chamber. And just like the name suggests, it actually has its own individual ventilation system. And so at the time of the study, it was really unclear to what extent the use of IVCs actually affected behavioral outcomes in rodents. So something else that's good to understand when we're talking about IVCs is that they can be held under two different pressure conditions. So under positive pressure, the internal cage pressure is higher than that of the surrounding room. Air is forced in through the ventilation system and can passively flow out. And negative air pressure is just the opposite. So air can passively flow into the cage, and it actually is forced out through the ventilation system. So part of the reason that there was kind of this big push to start using IVC systems is that they did have a lot of benefits, including a decreased cost, space, and number of cage cleanings that are required. They reduce the possibility of airborne disease outbreak in rats and also protect staff from aeroallergen exposure. And finally, they also prevent the buildup of volatile organic compounds like ammonia and carbon dioxide. That being said, a lot of the early research that was done that used IVCs did note some behavioral abnormalities, including an increased sensitivity to drugs, altered consumption of food and water, altered behavioral phenotypes, and higher overall anxiety levels. And so this last point, the higher overall anxiety, this was something that we really wanted to kind of research further, because since laboratory animals spend really a great deal of time in their own home cage, if that environment is anxiety inducing, then it would be a source of chronic stress, which would have a significant impact on research outcomes. So one way to kind of counteract anxiety is through the use of social housing. So a lot of research has shown that social isolation causes both the dysfunction of neurotransmitters and neural hormone dysregulation. And previous studies have also indicated that social housing typically reduces stress and anxiety, a term that's often coined as social buffering. But again, it was really unclear if the protective effects of social housing would still occur if animals were pair housed in an IVC system. And finally, we also decided to look at the oxytocin system for this study. So oxytocin is released from the supraoptic nuclei and the paraventricular nuclei of the hypothalamus in response to stressful environmental stimuli. And so it's also important to note that oxytocin is actually a self-regulating system. So the more oxytocin that's released means that more is actually being produced as well. And so social contact has been shown to release oxytocin. So what that means is that pair housing animals that will generally increase their social contact, and that will typically result in an upregulation of the oxytocin system. But once again, all of these studies have been done in OTCs, and so it was unclear if this would still be the same in an IVC system. So the study design, we took 40 male Long Evans rats and split them into 4 housing conditions. So 10 were housed in pairs under positive pressure, 10 were housed in pairs under negative pressure, 10 were housed alone under positive pressure, and the last 10 were housed alone under negative pressure. And then after spending 8 weeks acclimatizing to their respective housing conditions, all of the rats underwent the shock probe burying test. So the shock probe burying chamber is really just a plexiglass cube with an open top. And then a small electrified probe is actually inserted into the chamber. And then when the rat interacts with the probe, they receive a mild electrical shock. And so kind of the natural defensive behavior that this elicits is that the rat actually tries to bury the probe. And so defensive burying is the term that's used to describe when the rat takes its front paws and actually begins to push the wood chip bedding towards the probe. And so a lot of previous studies have indicated that higher levels of defensive burying typically correlate with higher levels of anxiety. We also measured the number of stretch attend postures. And so a stretch attend is when the rat stretches its whole body towards the perceived threat. In this case, it would be the probe. And then they quickly withdraw and retreat while keeping their eyes kind of locked on that probe. We also measured the number of rears, or the number of times that the rat stood up on its hind paws. And so this is important to measure because typically when a rat's faced with a threat, it's going to have increased levels of defensive behaviors like burying and like stretch attends. But rearing actually doesn't have a defensive purpose. So when faced with a threat, non-defensive behaviors are typically suppressed. And finally, the last two measures there, the number of contact-induced shocks and the score of each behavioral reaction to each shock. The last two measures are really just used to kind of control for confounds. So it's pretty logical to assume that if a rat gets shocked a lot more, that would also cause higher levels of anxiety, which would also cause higher levels of burying. So the goal of kind of taking the measurements of these defensive behaviors is that we want it to be not due to the effects of simply interacting with the probe itself. So I had two main predictions for this study. First, that pair-housed rats would be less anxious as indexed by the shock probe burying test compared to the single-housed rats. And secondly, that pair-housed rats would also have a higher density of hypothalamic oxytocin-producing cells compared to single-housed rats. I also did compare positive and negative air pressure, but because there hasn't really been an existing body of literature that's compared anxiety in terms of positive and negative air pressures in IVCs, I couldn't really make a directional prediction. So here are the results from the shock probe burying test. So you can see from the graphs that for the three main measures, burying, stretch attends, and rearing, there weren't actually differences between positive and negative air pressure. However, the pair-housed animals did bury significantly more. They did have significantly higher levels of stretch attend postures, and they had significantly lower levels of rearing. So this does suggest that the pair-housed animals actually had higher levels of these kind of classic defensive behaviors. And so this is really surprising because in the prevailing literature, pair-housing animals typically reduces their anxiety levels, and that's typically reflected in a decrease in defensive burying. Furthermore, to make it just that little bit extra weird, the single-housed animals buried not only less than pair-housed animals, but actually when we compared the levels of the single-housed animals to those levels of defensive burying in all of the previous studies, they actually had significantly lower burying levels on average. So something to keep in mind with these kind of strange results is that all of the previous literature was done using an OTC system, while we used an IVC system. And so keep these kind of surprising results in mind. I will be coming back to them. So after the behavioral results, we conducted an immunohistochemical analysis. So the rat brain tissue was processed to label oxytocin-producing cells in the hypothalamus. And then at that point, we're actually able to quantify the number of oxytocin-producing cells using a light microscope. So you can see here right away that the pair-housed animals and the single-housed animals under positive pressure, those three groups didn't actually significantly differ from each other. However, the single-housed negative pressure animals did see a significant decrease in oxytocin-labeled cells. And so something that's interesting to note from the literature is that chronic stress can actually cause the suppression of oxytocin production. So then it could be that the single-housed negative pressure animals were under chronic stress. And if that is the case, then that kind of begs the question, does this mean that the pair-housed negative pressure animals, them being paired with a conspecific, did that have a protective effect that prevented a similar decrease in oxytocin-producing cells? And kind of to sum up all of the results that we got, firstly, just to reiterate, our findings weren't consistent with our initial hypotheses. And so as I kind of got into on the previous slide, chronic stress is one explanation that could explain why the single-housed negative pressure animals saw a decrease in oxytocin producing cells. And what's also important to note is that the literature suggests that not only can chronic stress cause this decrease, but it specifically does that in the parvocellular regions of the PVN, which was the exact region that I was investigating. So then to kind of wrap it all up, we kind of took a look and said, okay, so we've come up with this hypothesis of chronic stress. Can we kind of use chronic stress to explain the results of the shock probe bearing test? And as it turns out, yes, we can. And it relates to the idea of active and passive coping strategies. So an active coping strategy is one where an animal kind of directly deals or handles the threat in front of it. And a passive coping strategy is one where it actually just actively avoids the problem. And so while previous studies have indicated that acute levels of stress cause an increase in defensive bearing, chronic stress actually causes a decrease in defensive bearing. And it does that through the suppression of active coping strategies. So animals under chronic stress tend to stop using active coping strategies and begin to use more passive coping strategies. And so why that's important is because then the pair-housed animals, it actually would suggest that their higher levels of defensive behaviors, if the IVCs were a source of chronic stress, was actually adaptive and they were showing an appropriate kind of defensive response, which would suggest that being pair-housed actually did have a sort of protective effect. So while the results of this study, to kind of sum up, they don't provide conclusive evidence of the effects that IVCs have on rodents, the results do somewhat suggest that IVCs themselves may actually be a source of chronic stress and that they should be investigated further so that we can kind of get to the bottom of it. And that's all that I have for you today. And if there's any questions, I'll take them now.
Thank you for the very interesting talk. Let's see if we have any questions. Yes. So while we're waiting, I'm wondering, you've all shown the effects of social housing, but from my perspective, I don't know if they're familiar with the rats that they're housed with. So I don't know if that will play an important role in this. So if I have, if I'm not familiar with the odor of my roommate, right, then being housed together would induce more anxiety. So is there any familiarizing like process in this?
Yeah, definitely. So that's actually a big part of the reason that we assign them the housing conditions and then we kind of leave them be for 8 weeks, because initially being paired with kind of another conspecific, that actually could be somewhat stressful. So the fact that we gave them 8 weeks was in a big part to kind of account for that, to make sure that kind of having a buddy in the same cage would actually be more of a reassurance as opposed to kind of another source of anxiety.
That's very interesting. Do you think if you record it for a longer period of time or house them together for a longer period of time, some of the effects may emerge?
Yeah, I think that's definitely a possibility. I think that we definitely found an effect for social housing. I kind of believe that we did find something, but I definitely think that if we kind of left them longer, it could definitely be stronger. Personally, I'd also kind of like to examine it if we started, we only use male rats. So what happens if we do similar things in IVCs, but we have males and females together, or we have siblings held together? Because I think both of those things would also kind of change up what we find.
Yeah, that's very interesting. I think the idea of, I guess the odor or pheromone, whatever, will really be depending on the relatedness genetically between the rodents. That would be
for
really
sure.
interesting. And I can see how this could maybe be a more, instead of just, this is right now as a basic science of the important part of your scientific procedure might be causing some effects. But I also feel like this could be something more like a scientific study, the effects of social isolation in this quarantine time. But yes, I feel their stress of being in the individually ventilated cages. And all right, it looks like we do not have any question, and we have come to the did I already answer? Oh, there is a comments from previous talk. It would be very helpful to see the data plotted as a scatter plot to see individual variability. This was for the, I think it applies to all of you. It would be interesting to see, yeah, maybe there are more social rats and then some are more introvert rats. That's very interesting. All right, I want to thank all the speakers. Thank you so much for the interesting talk. This is a very